In Chapter 4, we're going to learn all about the characteristic growth of bacterial cells. We're going to focus on bacteria in the lab, so under typically optimum conditions. But we will talk about how bacteria also grow in nature when conditions aren't always perfect. Bacteria reproduce via binary fission, and what this means is if you have one cell, every time the bacteria divide, they'll produce double the amount. So 1 would become 2, 2 would become 4, 4 would become 8. So they have this exponential growth that goes on. This is what happens typically in laboratory conditions. Obviously, they grow nice and rapidly. The generation time is the time that it takes to double the population. Now, different bacterial species have different doubling times or generation times. Some bacteria can replicate in just 20 minutes, while others might take 20 hours. The time it takes also will depend on the environmental conditions. If you give everything they need, they're going to replicate much faster than if they are in an environment that's not as favorable. So when a prokaryotic cell divides via binary fission, the consequences can potentially be problematic. So let's just think about bacteria growing in food that you're going to eat. If you start out with 10 bacterial cells that are pathogenic, and the cells are able to grow because they're inside the salad, so the environment is relatively nice, you can end up with 40,000 or 50,000 almost in just a few hours. So the question is, how can we figure out how long this takes? Well, the answer, you can use this equation. NT equals n sub 0 times 2 to the nth. So NT is the number of cells in a population at some given time. N sub 0 is the original number of cells in a population. And then the tiny little n is the number of divisions that have occurred. So the example was you had pathogens in a salad. You start out with 10 cells, so 10 cells would be the n sub 0. If we assume a 20-minute generation time, since it's 4 hours, we'd have 12 replications or 12 divisions. So, and we figured this out because 20 minutes goes into 60 minutes 3 times, so there's 3 divisions per hour, and there's 4 hours, so 3 times 4 is 12. This is sometimes confusing, and I totally get it. So if my explaining it here doesn't make you understand, come talk to me or ask me in class. I'll probably bring this up anyways. So now we know that NT equals 10 times 2 to the 12th. So if we do 2 to the 12th, we get 4,096. So NT equals 10 times 4,096, which means NT equals 40,960 cells that are found in your potato salad after 4 hours of sitting out. Makes you want to think about eating that food that you set out at a picnic, right? Not me though, because I'll just eat it. I've done it all my life. So the likelihood of all of a sudden those same bacteria causing me all this problem, I don't think so. I'm going to stick with what I do. So here's another question that you can look at. So let's assume that you are cutting chicken on your plastic cutting board and you set the chicken or the plastic cutting board into your sink. After 6 hours, you come back to clean your cutting board and there are 2.6 million bacterial cells on that cutting board. How many cells were originally there if the generation time of Salmonella is 30 minutes? We can use the same equation. In this case, we have the general population of the cells, 2.6 million. We don't know the initial population, but we know that there are 12 divisions because we had the Salmonella sitting there for 6 hours and the Salmonella doubling time is 30 minutes. So that means every hour there was 2 divisions, so 2 times 6 is 12. So the equation would look like this: 2.6 million equals n sub 0 times 2 to the 12th. Another way to put that would be 2.6 million divided by 4,096, because you're going to take this number, which is 4,096, and divide it on both sides to get n sub 0 alone, and you end up with n sub 0 equaling approximately 635 cells in the population initially. So in nature, bacteria don't always have the best conditions where they're going to grow most effectively. And so bacteria often will form associations with other cells or communities with other cells. What this is termed is a biofilm. And I think I mentioned biofilm before, but biofilms are the gunk that you find on your teeth every morning and every evening, which is why you brush your teeth twice daily at least. It's the stuff that can build up on your tub if you don't wash your tub. It's the slimy stuff that's found on rocks when you're walking by a pond or by a lake. That slimy material, that's a biofilm. And it just protects the bacteria. They grow in a group and then they're less likely to be damaged by any type of chemicals. So how do we form a biofilm? We start with bacterial cells just binding to some surface. The bacterial cells that bind start producing a chemical that helps them to stick to the surface. The initial attachment is reversible, but once other bacterial cells move in, that initial attachment is no longer reversible. So other cells come in. It doesn't have to be the same types of cells. You can have different types of cells that move in, and then they start replicating and they produce different waste products that can be used by the other cells. So the cells at the bottom might produce some type of waste that the cells above can utilize. Cells at the above produce some waste that the cells at the bottom can utilize. They have little channels within the biofilm so materials can move throughout the biofilm. This allows them to talk to each other as well. The only cells that are going to be potentially damaged by chemicals are going to be those cells at the very surface of the biofilm. And so here we have bacteria communicating. So quorum sensing is a form of communication within the biofilm and between cells in an environment, allowing the bacteria to know what's going on, how many cells are in the environment. And antibiotics and other chemicals don't tend to hurt or harm biofilms. They're not going to cause any problems. So what typically happens is if you have a biofilm, say biofilm on your teeth or biofilm on some medical equipment, the only mechanism is to mechanically remove the biofilm. So this brings us to the importance of biofilms. Biofilms can be very dangerous. They can lead to gum disease, or they can cause people to become septic if there's some type of mechanism inside that has a biofilm on them. So like I always think of a pacemaker. If a pacemaker has a biofilm that formed on it, then that individual is going to be very septic until we get that biofilm removed, and that can lead to death. Biofilms can form in pipes and drains, and that can lead to blockages. And of course, I told you that biofilms help microbes to be much more resistant to any chemicals. So the only mechanism of removing a biofilm is mechanical removal, which is why we brush our teeth every single day, at least twice a day. Biofilms can be very beneficial, though. We use biofilms in our water waste treatment. They actually help to filter our water. So we use biofilms and filter water through to produce fresh water from wastewater. Bioremediation, we actually use biofilms to clean up oil spills and other materials as well. Now we're going to switch focus, and we're going to talk about laboratory conditions. So these are the conditions that we put our bacteria in because we want them to grow effectively. So the first thing we'll talk about is how we produce a pure culture. So when we grow bacteria in a lab, we give the bacteria all the things that they need to grow so they'll grow really effectively. But if we're growing bacteria in a lab, we have to be able to produce what we call a pure culture. So we only have one species growing, so we can run tests on those species. One way to do this is we use a process called aseptic technique. Aseptic technique allows us to decrease the potential for contamination of bacteria. And then we can grow the bacteria on a liquid or a solid culture. And so over here I have Serratia marcescens. She's a beautiful species, producing in the right conditions pink colonies. So you can see on an agar slant and in an agar plate. But here's another agar slant where she's white. And the reason for this is at room temperature, she turns pink. But at 37 degrees Celsius, she'll stay white, or this, white color. So here's nutrient broth that she's growing in, and here she's pink because she was in room temperature, and here she's that whitish color again. So if we want to produce pure cultures, we need to have a certain type of culture medium that allows bacteria to grow on a solid surface. We had initially liquid material, so the liquid was called nutrient broth, and bacteria grow, but it's really hard to isolate single cells in a broth. So we found a solidifying agent in red algae, actually, that is called agar, and we use that agar to cause the broth to solidify. So over 95 degrees Celsius, it will liquefy, and so when we sterilize the agar in the autoclave, it actually becomes a liquid. and then it solidifies when it hits 45 degrees Celsius or below. And so we pour our agar into plates after it has cooled down a little bit. We pour the agar into little plastic plates called Petri plates, and then we allow the agar to solidify and then flip them upside down. And we are able to produce individual colonies. And so there's an example of an agar plate with individual colonies. In our lab, we're going to learn how to separate bacteria using a process called the streak plate method. This is a very easy method, but it takes practice. So don't be upset if it takes you a couple of times. The idea behind it is we're going to start out with, millions of cells and hopefully cut down the number of cells to just a few cells. And so the first thing we're going to do is we sterilize our loop. When you sterilize your loop, you want to sterilize for about 20 seconds. And I know it's only showing the loop itself, but you want to sterilize that entire needle area, okay? And then after the 20 seconds, you're going to take that sterile loop and you're going to dip it into a broth. That broth could have, 2, 3, 4 different species of bacteria growing, depending on where you're getting the broth from. Say this is from a sputum sample from a patient that you're working with. You then take that broth, that loop, I should say, and you streak it over one small section of the plate. We then re-sterilize the loop for that 20-second period. And then we're going to pull bacteria from this first section to the second section. We only pull one time, maybe 2 times, and then we just streak that second section. We then re-sterilize the loop, 20 seconds, and then we grab bacteria from the second section and pull to the third section and streak that area. Re-sterilize the loop and pull bacteria from the third to the fourth section. And this is what you'll end up having. And again, I use Serratia because I think Serratia is really pretty. So here's that first section, lots and lots of bacteria. Second section, still a lot of bacteria. Third section, you're starting to see some separation. By the fourth section, you get those individual colonies out here. So how do bacteria grow in a lab? They go through a process that is typical of all bacterial cells in optimum conditions, called the growth curve. And they have this phase called the lag phase, which is this period here. Lag phase is when the bacteria are getting used to the new environment. So when bacteria are transferred from one medium to another, it takes them a little bit of time to acclimate to the new conditions. And then they're going to start replicating. And so this is where the cells are dividing, which means DNA is replicating, proteins are being produced. And so this is also where the bacteria are most sensitive to different antibiotics. Because if you need proteins, antibiotics that inhibit protein synthesis will stop the production of those proteins and the cells can't divide. Think of penicillin inhibits the production of peptidoglycan. So the cells won't be able to produce the peptidoglycan and then can't survive in the environmental conditions and so can't replicate. Once we hit that log phase, or once we have replicated multiple cells, we're then going to hit a phase called the stationary phase, where bacteria might still be growing, but at a much slower rate. And some of those cells are going to be dying because they don't have the nutrients they need. But those that die can actually give the nutrients to the other cells who can replicate. The last phase is called the death phase, where the number of cells that are viable and can replicate will actually decrease. And so decreasing, or the death phase, is also exponential, but it's a much slower exponential rate than the log phase. I'm going to stop here and I'll pick up on the next lecture.